right, so welcome to the third segment of Chapter 17. We're going to be talking about translation, which is the final process, or almost the final process of gene expression. But we're going to kind of talk about protein folding and the localization of proteins as well. So let's talk about translation first. So translation is basically the use of RNA, or it's RNA directed, and it involves the synthesis of a protein or a polypeptide. So genetic information at this point has already flown from the DNA to the RNA, and now we have the messenger RNA, and translation involves the flow of this information from the messenger RNA to produce the protein. And again, this is called translation. Now, you could say that translation is RNA-directed synthesis of proteins, or a polypeptide. Now, the components of translation involve several different types of RNAs. So the cell is going to translate a code that's in the messenger RNA. So, of course, messenger RNA is involved. And this code is going to be used to generate a protein. And this is done with the help of what's called transfer RNA, or tRNA. Transfer RNAs have amino acids attached to them, so they're going to transfer the amino acids to the growing polypeptide, which is occurring at the ribosome. Translation is a complex process in terms of how the mechanics of it, and of course the biochemistry of it as well. Your textbook goes over the brief details of it. Now let's look at the process here, or what's involved. So again, like I said, the messenger RNA is involved, and that's shown in red here. The ribosomes, of course, are involved because those actually catalyze the chemical reactions to some degree. And these candle-like structures, which are shown in your textbook, represent the transfer RNAs. And, of course, the growing peptide has amino acids connected to each other, and they're connected by peptide bonds. So remember, the transfer RNA, like I said, it has amino acids attached to them. So each transfer RNA is going to have one amino acid attached to it. And it's attached at the 3' end, where the wick looks like, in reference to the candle, where the wick is. Let's look at the actual structure of a transfer RNA. Now, a transfer RNA first has what's called the anticodon. So remember the codons in that codon table? Well, the anticodon is basically the reverse complement of the codon. And this allows the transfer RNA to recognize a codon and actually bind to it by hydrogen bonding. So that's how the transfer RNA basically delivers the correct amino acid, because it has that anticodon which matches up with the codon. Now, the structure of the transfer RNA is quite complex. It is RNA, so it's single-stranded. However, it can fold upon itself and create these loop-like structures. And, of course, hydrogen bonding between the bases help form this three-dimensional structure. Now, this structure does not look three-dimensional because it's on flat paper, and it's just showing the straight base sequence of it. You can see where the 3' end and where the 5' end is. They're both on the same end. However, it's the 3' end that the amino acid is attached to. On the opposite end is a loop containing the anticodon. So this anticodon is read in this way, AAG. Well, it binds to the codon, which is in the messenger RNA, which would be CUU. So you can see they kind of, they obey this Chargaff's rule and the hydrogen bonding rules, where adenine binds to thymine, or uracil, and guanine binds to cytosine. Now, looking at the three-dimensional structure, you can see this is kind of a ribbon diagram. You can see the hydrogen bonding that are occurring between the bases, and, of course, the anticodon end and the 3' end, which connects with the 5' end. However, remember that 3' end is the end where the amino acid is attached. So looking at this actual ribbon diagram and seeing this three-dimensional structure, you can kind of relate it to what's shown in your textbook. So in your textbook, again, it looks like a candle. on the bottom of the candle, the base of it, contains the anticodon. You can see that would be represented here in the actual three-dimensional structure of the transfer RNA. And on the 3' end is shown, it looks like a wick, okay, if we were going to refer to the candle. And again, this is going to be where the amino acids are attached. Now, this shows one that an amino acid hasn't been attached yet. Well, how do the amino acids get attached to the transfer RNA? Well, we're going to talk about that. Translation requires two accurate steps. Okay, one of them is a correct match between the transfer RNA and its amino acid. This is done by the enzyme called amino acyl transfer RNA synthetase. The second step that needs to occur accurately is a correct match between the transfer RNA anticodon and the codon, which is in the messenger RNA, and this is done by hydrogen bonding. Now, there is some flexible base pairing at the third base within the codon. This is called the wobble base. It allows some transfer RNAs to bind more than one codon. Now, let's go back and look at the amino acyl transfer RNA synthetase enzyme and how it works. So this structure right here, it's purplish blue, I guess, it represents the enzyme, and the enzyme is called amino acyl transfer RNA synthetase. And again, an enzyme is a protein, so it's a protein that catalyzes a chemical reaction. What is the chemical reaction? It's the connection of an amino acid to its transfer RNA. So we have amino acyl transfer RNA synthetase, and it has two binding sites. One of those for the amino acid. You can see the amino acid is going to enter here. And then, of course, during the process, a phosphorylated intermediate needs to be produced, so hydrolysis of ATP occurs. So the process of it's called charging in a transfer RNA, requires energy in order to proceed. Now, you can see here that we have the amino acid, which is bound to its binding site, and you can see the ATP has been hydrolyzed and a phosphate has been added to the amino acid. Now, this creates a phosphorylated intermediate. Remember, phosphorylated intermediates make molecules a lot more reactive. So this is going to make this amino acid much more reactive. We want it to react with the transfer RNA. So the next step, of course, is where the transfer RNA binds into the binding site for transfer RNA. Now, this shows an actual space-filling model and a stick model for the transfer RNA. So the space-filling model represents the enzyme, and the stick model represents the transfer RNA. So this is a computer-generated model based on predicted structure, three-dimensional structure for each of these. And you can see the space where the amino acid is attached. So if we look at the cartoon picture, which is the one shown in your textbook, where we can see that the amino acid has been added onto the 3' end of the transfer RNA, and of course the phosphate is going to be released. So the phosphate is there to help it, do that reaction. And then, of course, once the reaction is done, the enzyme is going to go back to its original structure. Remember, it's just a catalyst, and it'll kind of recycle, and it'll be able to continue the cycle over and over again. But once it releases the transfer RNA and the amino acid, which is attached to it, it is now called a charged transfer RNA. Now, ribosomes are really the site where the peptide synthesis occurs. So ribosomes and their structure facilitates the coupling of the transfer RNA, the anticodons in it, with the codons in the messenger RNA. Now, there are two subunits of ribosomes. There's a small subunit and a large subunit. And of course, it's made up of ribosomal RNA and proteins, ribosomal proteins. So it makes this overall functional ribozyme is really what it's called. It's an enzyme. It acts like an enzyme. So let's look at the structure of the ribosome. So we can see the ribosome here. This is basically a computer-generated space-filling model of the ribosome showing three sites that are generated once the small subunit and the large subunit combine. You can see the messenger RNA here and a growing peptide that is being generated as protein synthesis is occurring. So what's important here is that there are three sites that are ultimately generated when the two subunits combine together, and that's called the A site, the P site, and the E site. There are different things that occur in each of these sites. So the P site is really where the peptide is going to be growing, the peptide chain is going to grow and emerge out of the ribosome. The A site is going to basically be the site where the new transfer RNA enters. So the new transfer RNA is going to be the one that's holding on to the amino acid that is next on the polypeptide chain. And then the E site can be considered the exit site. So eventually, the amino acids will be removed from the transfer RNA, and then it'll be an uncharged transfer RNA. Well, the uncharged transfer RNA ends up in the E site, where it's going to exit the whole system, and then, of course, become recharged. Look at this process. Now, starting from the very beginning, we can see here the small subunit and the large subunit of the ribosome. And when they're kind of come together, and you can see that the sites are mainly in the large subunit. So we can see the A site, the P site, and the E site. So remember, the A site is going to hold the new transfer RNA, which is holding the next amino acid in the polypeptide chain. The P site is going to allow space. This is called an exit tunnel right here. So this is where the growing peptide is going to be emerging out of the ribosome during protein synthesis. And then the E site is going to be where the uncharged transfer RNA is going to enter, and then it'll eventually exit the site. Now, adding some things to this, we can see that the messenger RNA is kind of sandwiched between the small subunit and the large subunit. And here we can see some transfer RNAs in here. Here, this was in the E site. This is an uncharged transfer RNA. So the amino acid that was attached to this uncharged transfer RNA was this one right here. Okay, so, or actually it was the triangle. So the triangle was attached to the circle, which happened to be in the A site when it first entered. Either way, this is an uncharged transfer RNA, and it exits the system. So you can see the A site is holding on to a charged transfer RNA, which happens to hold the next amino acid in the polypeptide chain. And then this we can see is the growing polypeptide chain. Eventually, this circle amino acid is going to be attached to this end of the diamond or square amino acid, and then the whole peptide will be in the center. We'll kind of look at this process as we go through. So, polypeptide synthesis occurs in 3 stages, and if you remember translation or transcription has the same 3 stages. However, different things are involved and different things occur. So there is an initiation stage, an elongation, and a termination stage. Now, just like transcription, there are factors which help with the process. Well, in this case, instead of calling them transcription factors, they are also called translation factors. So these are going to be special proteins and enzymes which help the process of translation. So the initiation stage is basically the stage which helps produce the initiation complex. Okay, so this involves the formation of the initiation complex. So let's talk about how it's done. So if we look to the left here, we can see there is a messenger RNA, and we have a small subunit. So the small subunit is going to bind to the messenger RNA. It kind of recognizes that 5' cap on the messenger RNA, and then it's going to scan for the start codon, which is AUG, and you can see that up here. You can see the red is the AUG. So once it scans and it pauses where the AUG is, this allows the transfer RNA, which is actually called the initiator transfer RNA. There's really only one because AUG is always the start codon, so then this initiator transfer RNA is always going to be the one that's going to bind to the start codon because it has the corresponding anticodon. So you can see AUG here, and then the transfer RNA is going to be CAU. Okay, and then of course, since AUG, remember the codon table, says that it codes for the amino acid methionine, of course, the transfer RNA, which has the anticodon to AUG, is going to be connected to the amino acid methionine. Okay, so once that happens, then we need some energy to complete the process. The energy in this case is not coming from the hydrolysis of the ATP, it's coming from the hydrolysis of GTP. So GTP is going to be hydrolyzed into GDP and inorganic phosphate. Now we have some energy in order to attach the large subunit and create the initiation complex. So here we can see the initiation complex has been created. Once the initiation complex is created, then elongation can occur. So let's talk about elongation. Now, elongation is the process of adding amino acids to the peptide to create the entire polypeptide. The elongation stage, or translation in general, is going to proceed along the messenger RNA in the 5' to 3' direction. So let's look at this process. So here, this has already been occurring, but we're going to talk about the cycle in general. So we have the messenger RNA here. We have a new transfer RNA entered, okay, and then we have a growing peptide in the P site. Now, the E site does not show an uncharged transfer RNA because it's already left the system. So let's talk about the 3 stages of elongation. The first stage is called codon recognition. So codon recognition involves where the next transfer RNA, which has the correct and corresponding anticodon, which matches with the codon that's in the messenger RNA. So it's recognized by, favorable base pairing, hydrogen bonding. And, of course, it's going to bind. Of course, we need energy. And, again, all the way through this process, instead of the hydrolysis of ATP, it's the hydrolysis of GTP is going to be producing or providing the energy for this process. Okay, so that's the first step. The second step is going to be involving the peptide bond formation. So you can see that's kind of starting here. So you can see the growing peptide, and the last amino acid that was added to the growing peptide is this circle. Well, now the circle is going to be attached to the diamond, which is the next amino acid. So you can see that occurred right here. That is the formation of the peptide bond. Okay, so a peptide bond occurred between the circle and this diamond, which was the new amino acid being attached to the polypeptide. Now, at the same time, even though it's not showing this, the ribosome is going to scan along the messenger RNA to the right. Okay, so it's going to move to the right. So what's going to happen is, this is called translocation, and of course energy is required for this process. What's going to happen is this transfer RNA that was in the P site, which just lost to the growing peptide, to the new transfer RNA, is now going to be considered uncharged, and it's going to exit the E site. Well, it ends up in the E site after translocation occurs, and then it leaves. Now we have the growing peptide here, and we have A empty. Okay, so site A is now empty, ready for the next transfer RNA to recognize its codon, which is on the messenger RNA in that space now. Okay, so we have those 3 steps. We have codon recognition, we have peptide bond formation, and translocation. That's going to continue to occur in a cycle until the ribosome reaches a stop codon. And this is where termination occurs. So termination is going to occur when the ribosome and the whole system reaches a stop codon, and the stop codon happens to be in the A site. Now, once the ribosome reaches the stop codon, this induces basically attracts the release factor. So the release factor is going to enter the A site along with a water molecule. Okay, this is the start of the breaking up of this whole system. Okay, so once the releaser enters, then the peptide is going to be released. Okay, so now we have the transfer RNA, which was holding the polypeptide. Well, that's going to be cleaved off, leaving the polypeptide free to float around in the cytosol. And now we have an uncharged transfer RNA. Then energy is going to be required to basically remove the ribosome from the messenger RNA so that the ribosome can then attach to a new messenger RNA and produce another protein. Okay, so this is the whole process of the end of translation, right, the termination. So let's talk about what happens after and or during this process. So what can also happen is what's called polyribosomes, or a polysome. So this is where many different ribosomes can translate the same messenger RNA simultaneously. So they're going to be positioned along the messenger RNA, continuing to scan along the messenger RNA, producing that protein. So this allows the cell to produce many, many, many copies of the same protein, especially if it's needed very quickly. Now, let's look at we have a electron micrograph of a polyribosome, right? So you can see it's like a beads on a string, like we mentioned before with the chromosomes and the histones. Well, except these are ribosomes on the messenger RNA. And your image that's shown in your textbook shows a cartoon image of this. So this is basically a bunch of ribosomes along the same messenger RNA producing the same protein pretty much simultaneously. So again, many ribosomes is called basically polyribosomes or a polysome, which is basically a messenger RNA with multiple ribosomes along the length of it, and this is meant to produce a protein in very high quantities relatively quickly. Now, once translation is completed, that might not be enough for the protein to be functional. So some proteins need to be modified after translation occurs. So this is called post-translational modifications. Now there's many different types of protein modifications. However, your textbook only goes over a specific one. So during and after the synthesis of a polypeptide, it's starting to twist and turn and starting to fold into its functional form. Now, it may not be able to fold into its complete functional form until part of it might need to be cleaved off. Okay, so again, one post-translational modification might be the cleaving of a section of the protein so that it can fold correctly. Now, some polypeptides are going to be activated by this cleaving. Some of them might be activated by the binding of another protein, or perhaps the addition of a methyl group or another type of functional group might be required in order for it to be functional, or even a cofactor. So let's say it needs a cofactor. So there's a number of different modifications to proteins that may be required for them to be completely functional. Now, going into the targeting and the localization of proteins, and of course overlapping with modifications, let's talk about this for a moment. So there are two types of ribosomes. Remember, the free ribosomes and the bound ribosomes. So remember, the free ribosomes are floating around in the cytosol, and then you have the bound ribosomes, which are actually attached to the ER. Well, most bound ribosomes, or all of the bound ribosomes, were free at one point. Okay, so free ribosomes are mostly going to be synthesizing proteins that have a function in the cytosol or may be relocalized to the nucleus. The bound ribosomes, again, were once free ribosomes. However, they're producing a protein that needs to go to the ER. So since it needs to go to the ER, it's going to carry it to the ER as it's being produced. So it makes it a little bit more efficient. So now ribosomes can, obviously, just from talking about this, the ribosomes can switch from free to bound or bound to free. So they can switch between these two forms. It depends on what protein they're producing. All right, so peptide synthesis always occurs in the cytosol. Okay, so it's always going to occur in the cytosol. Whether it goes to the ER, it's still occurring in the cytosol. So synthesis finishes in the cytosol unless the polypeptide is signaled in some way to go somewhere else, such as the ER. So we're going to talk about signaling of peptides to the ER. Now, looking at this process here, let's start with step number 1. This might look a little bit complicated. So first of all, let's kind of orient ourselves. So we have the ER lumen, okay, so that's the space inside the ER. This is the membrane that makes up the ER, and then, of course, the pinkish area is going to be the cytosol. So again, the protein is being made in the cytosol. Look, you have the red, which is the messenger RNA. You have the ribosome attached to it, and the peptide is growing out of the ribosome. Now, this peptide happens to be signaled, or it has a sequence which directs it to the ER. So it's a protein that needs to go to the ER. So what's translated with it is called a signal peptide. So it's just a small section of amino acids, right? Sequence of amino acids, which is recognized by what's called the signal recognition particle, SRP. This is a protein, right? So the signal recognition particle has a space that is meant to bind to this signal peptide. So since the signal peptide is present, the signal recognition particle is going to bind to it. Now, once the signal recognition particle binds to it, the signal recognition particle is going to direct, basically it's attracted to a receptor, which is the signal recognition particle receptor protein on the surface of the membrane of the ER. So that's going to bind to that protein, okay? Once it binds to that protein, okay, this is going to initiate a translocation complex to open up. Okay, so this basically creates a channel. So once it binds, this is going to open up, and it's going to create a channel so that the protein, as it's being produced, it's going to enter the ER. So you can see it entering the ER here. And you can see that the signal peptide is still bound to the signal recognition particle, which is attached to this whole complex right here. Now, eventually, the signal peptide, which is bound to the signal recognition particle, is going to be cleaved. So it's going to be cut off the protein. Okay, so it's that signal peptide which directed it to be located or translocated to the ER. Now we have the peptide in the ER. And then whatever happens to it after that is what's supposed to happen to it. Okay, so that's a way of localizing proteins to their particular functional location. Now let's talk about a little bit something different. So remember, we've been talking about gene expression, and in reference to really eukaryotes, because remember, eukaryotes have the ER. There's a lot more. It's a lot more complex. Now, archaea are also prokaryotes. However, they're a little bit more closely related to eukaryotes. Okay, so they share a lot of features with eukaryotes, and some of those features involve the process of gene expression. Okay, so let's compare the gene expression in bacteria, archaea, and eukarya. Now, first comparing the bacteria with the eukarya. Remember, bacteria are prokaryotes. Eukarya, of course, are eukaryotes. They're going to differ in a lot more things than any differences that might be between eukarya and archaea. They definitely differ in their RNA polymerases, the termination of transcription, which we talked about already, and the ribosomes and the size and structure of the ribosomes. Archaea tend to resemble, or they're going to be more closely related to the eukarya in all of these aspects. Bacteria can simultaneously transcribe and translate the same gene. They don't have a nucleus, so you can imagine transcription and translation basically occur in the same place. So they can simultaneously both transcribe and translate genes. In eukarya, transcription and translation, of course, are separated because there's a nucleus, and they're separated by the nuclear envelope. Remember, transcription occurs in the nucleus, translation occurs in the cytosol. In archaea, transcription and translation are likely coupled. Again, they do not have a nucleus either, but as far as I know, or that's at least been mentioned in your textbook, that they're likely coupled. However, it's not known for sure. Now let's talk about this simultaneous transcription and translation process. So here we can see that actually happen. We see beads on a string that represent the messenger RNA and the ribosomes. And all of these beads on a string are hanging off another string, which represents the DNA. So we can see the DNA. We have RNA polymerase, which is synthesizing the messenger RNA, which is the long string in which the ribosomes are attached to. So you can see polyribosomes. So this is part of what allows bacteria or prokaryotes to be able to replicate relatively quickly, because they're a lot more efficient at it, right? So they don't have a nucleus. So transcription and translation are not separated in any way. So they can occur simultaneously. And this is basically the cartoon image which represents this process. So we can see the DNA here. We see RNA polymerase scanning along the DNA to produce the messenger RNA, which is in red, and you can see the ribosomes attached to the messenger RNA, and then the proteins emerging from each of the ribosomes. Okay, so quite interesting how prokaryotes are relatively efficient. Now let's kind of revisit the thought of a gene. So we kind of talked about that, of course, in chapter 14 and 15. Now, the idea of a gene or what a gene is has changed or evolved throughout the history of the field of genetics. So it's been considered as just a discrete unit of inheritance at first, and then it kind of moved into a region representing a specific nucleotide sequence in a chromosome. But then later, it's now more like a DNA sequence that codes for a specific polypeptide chain. So remember, there are some DNA sequences that are specific regions on the chromosome. However, they may not code for a protein, right? So there's, remember, the untranslated regions and the non-coding regions in eukaryotes. So the best description would be this final one, right? So a gene could be described as a DNA sequence that codes for a. Okay, so in summary, combining all the more important things about a gene can be defined as, one, a region of DNA, okay, that can be expressed to produce a final functional product. Okay, so to be more specific, it doesn't have to be just a peptide. It could also be an RNA molecule. Remember, there are functional RNAs like the ribosomes, right? So they have a function. They catalyze chemical reactions like peptide bond formation during protein synthesis. So again, you must include the possibility that it may not be a protein. It could be a functional RNA as well. Let's go ahead and review this whole process of gene expression. So if you remember, in the nucleus, which is where the DNA is. So since the DNA is going to be in the nucleus, it makes sense that transcription is going to occur in the nucleus as well. So remember, the transcription in eukaryotes generates a pre-messenger RNA. Okay, in prokaryotes, it's automatically a messenger RNA, which can be translated. Remember, transcription and translation can occur simultaneously in prokaryotes. However, in eukaryotes, it's a lot more complex. So here we have the pre-messenger RNA. Remember, it's a pre-messenger RNA because it must be modified. So there's RNA processing. And RNA processing occurs in the nucleus because these things must occur to the RNA to create messenger RNA so it can leave the nucleus through the pores and go to the cytoplasm. So what ultimately happens, remember, we have the 5' guanine cap added to the 5' end. We have the 3' poly A tail added to the 3' end. And then introns are spliced out by the spliceosome. And then, of course, the exons are reconnected. And then we have a messenger RNA. So once the messenger RNA is formed, it's going to exit the nucleus and enter the cytoplasm. So this is going to be where, remember, the ribosome binds to the 5' end, and then it scans for that start codon, which is AUG, and then the large subunit of the ribosome and the initiator transfer RNA can bind, and then, of course, elongation occurs and the production of a peptide. Now, in order for all of this to occur, we need to have charged transfer RNAs. So remember that protein or that enzyme called aminoacyl transfer RNA synthetase. Okay, so aminoacyl transfer RNA synthetase is going to add each of the amino acids to the transfer RNA so that they can be charged and take part in protein synthesis. Okay, so translation is basically the reading or basically deciphering of the code which is in the messenger. Remember that codon table? That's messenger RNA codons. Okay, and remember the anticodons the transfer RNAs have the anticodons. So the anticodons are like being able to, decipher a secret code, right? So they have that ability. So they can tell us, the anticodons can tell us, and the codons can tell us what amino acid will be next, or the entire sequence of the amino acid. All right, so that's the whole process. Remember, after a peptide is made, it may need to have further modification, such as cleaving of its section. It might be directed to the ER or even the mitochondrion or some other location in the cell.